Well, good morning, everyone. I'll just get going here. So I did manage to catch the flight, which I guess is an accomplishment now, since we haven't always been able to do that. I'll have to kid my friend Richard Cummings about not getting out of Boston. But what I want to talk about today is a targeted approach, and you've heard a little bit about targeting specific proteins and glycoproteins that are made by human cells, but this would be an approach to determine specific aspects of the human genome. And since there are some non-glyco people in the audience, this is a little summary here of recent impact. Gordon talked about the structures of glycans on IgG regulating ADCC, and he also talked about pathogens being covered with glycans and the interactions of those pathogens with human cells. I'll just mention a couple of other recent impacts. So there are two drugs now that are in phase 3 trials. These are anti-inflammatory drugs, and they inhibit P-selectin glycan interaction. One is a small molecule, and the other is a monoclonal antibody that inhibits that interaction. These are in phase 3, and the disease that they're targeting is sickle cell syndrome. It's a vaso-occlusive disease. And basically, these reagents inhibit the interaction and really stop the inflammatory response within an hour or so. And so this will be we anticipate that these will be approved for sickle cell syndrome treatment. And then after that, more general inflammatory diseases will see treatment here. So again, knowing the glycans and knowing the proteins that interact with them is going to lead to 2 new drugs on the market. In addition, Glycomimetics has developed an E-selectin inhibitor, and this actually mobilizes bone marrow AML cells, chases them out of the bone marrow compartment, and causes them to become susceptible to gemcitabine. And so this is now in, I believe, phase 2 trials to increase the efficacy of chemotherapy for AML and for other leukemias as well. So these are a couple of big advancements in the field of taking glycobiology and applying it to human disease. And I'll also mention that there are well over 100 congenital disorders of glycosylation that have been identified. These are mutations, in the most part, in enzymes and proteins that are responsible for the biosynthesis of glycans that are on glycoproteins. And so these are, so we know specific mutations that cause changes in glycogene expression or mutations in proteins, and these give rise to congenital disorders. So again, showing that understanding glycans is extremely relevant to human disease. You've seen this cartoon before. So really what we're interested in is being able to describe the glycans that are on glycolipids, and in this case, I'm just showing glycoproteins, and identify ultimately where those glycans are expressed on proteins, so site-specific information. This would be the greatest goal of the Human Glycome Project, is to be able to define this relationship. And so, looking at glycomic strategies that many people in the audience employ, basically we can start with free glycans that are found, for example, in milk and other bodily fluids. But in most cases, we start with cells and sometimes tissues, where we prepare glycoproteins, trypsinize them so that we have glycopeptides, and then these can be separated and analyzed to give us information on glycans on specific proteins and the sites that they're on. And then, of course, we can release them and separate those glycans. Sometimes we tag them, sometimes they're permethylated and separated. And the idea is, once we take everything apart, then we can use bioinformatic tools to put everything back together again and understand which glycans are on which positions on which protein. So this would be the scheme, and obviously it's extremely complicated to be able to do this. So at the level of glycoproteins and glycopeptides and glycans, it would be very useful if we could target specific classes of glycans and use that to be able to pull out specific classes for analysis to simplify. And so that's really what I'm going to be talking about, a targeted approach using specific reagents like the antibodies that Gordon mentioned before, or lectins, or modified enzymes, which I'll talk about in just a second, to segregate glycan structures of interest. And so really this is basically using hooks to go in and pull out specific glycoproteins or glycopeptides or even glycans for analysis to simplify, so we can do a higher throughput and also more detail in understanding those. And hopefully it's not just a bare hook, it actually has some bait on it, and we can catch some really big fish. So I want to describe this by first talking about polysialic acid, which is the area of research that really sort of got us into this whole idea of targeting, and then expand that to talk about all sialic acid containing glycans. Could we actually define the sialome of a cell, for example, or a tissue? That is to say, all the glycosyl transferases and all the glycans that have sialic acid on them. You've heard about that before. And at the level of either peptides or sialoglycoproteins. And I'll give an example of using a targeted approach, not for sialic acid, but for other glycoproteins, using as the hook L-PHA, L-phytohemagglutinin, pulling out specific glycoproteins with beta-1,6 branched glycans on them in breast carcinoma. So just to show you how we progressed in this area, this is a list it's probably a little old now, of mouse and human glycan-related genes. This was put together by Kelly Moorman as part of our National Center for Biomedical Glycomics. And you can see around 1,000 genes encoding proteins that are either binding proteins or transferases, hydrolases, or related directly to glycan biosynthesis and breakdown, or so-called glycogenes. And so we began studying this in embryonic stem cells, actually starting out first with mouse. And Mike's going to talk a little bit later and go into more detail on the stem cells. But this was really our first attempt at this. And Kelly basically developed specific PCR reactions for all the glycogenes that I just mentioned. And we started out in mouse, as a matter of fact. And when we looked at the glycotranscriptome from the mouse embryonic stem cells and compared that to two differentiated cell types, embryoid bodies actually, which is a mixture of cells and extraembryonic endoderm, and these are the levels of transcript done by PCR comparing the three different cell types. and the line at the top is basically the differential. So you can see these are sialylotransferases here, for example, and you can see significant changes, but there were some really large changes that got our interest, and these were in enzymes that synthesize polysialic acid, which I'll describe more in just a second. And it was really not known that the differentiated products here showed a great increase in the transcription of these genes. And so we were very interested in determining, is the product really made? Though embryonic stem cells really have little or no expression, but these differentiated cells appeared to have a lot of transcripts making polysialic acid. So we got an antibody from Karen Colley, and then another antibody from Rita Girardi-Shan, who work on polysialic acid, and we stained the embryoid bodies, the differentiated cells, with that antibody, and you can see the embryonic stem cells don't stain at all. We have very, very strong staining of polysialic acid. So this was really not known at the time. Polysialic acid was really described first during late neuromorphogenesis as axons are trying to find their targets, and they express polysialic acid and then turn it off when they begin synapsing. But this is just very early in differentiation from embryonic stem cells into a product. And interestingly, when we looked at human embryonic stem cells and their differentiation into definitive endoderm, this basically is a marker for the stem cells, and here's the antibody to polysialic acid. And if we induce these cells to differentiate to definitive endoderm, and this is done in our center by Steve Dalton, who studies early embryonic differentiation in stem cells. And you can see nanog is turned off, but then polysialic acid is turned on very strongly. So for both mouse and human, polysialic acid is being expressed. And I'm not going to show you any data on experiments, further experiments along the lines of definitive endoderm, but I will now describe a little bit about polysialic acid. So sialic acid is linked alpha when it's found in glycoproteins and in glycolipids. And this would be a sialic acid here, linked alpha 2,3. As you'll see, sialic acid is normally in humans linked three different, there are three possible linkages. The polysialic acid is alpha 2,8 linked, and it is synthesized on a sialic acid that's non-alpha 2,8, and then this basically shows an N-link structure and another N-link structure. The polysialic acid was really discovered in bacteria named cholaminic acid, and Yucafine found it in rat brain in '82, and many researchers have studied this, and it can range from 8 to up to 400 monomers in mammals. So it's a very, very interesting structure. Its expression is really correlated well with cells that are going to become migratory, as a matter of fact. And the most common acceptor for it is NCAM, the neural cell adhesion molecule. And this is work by several investigators, including Karen Colley, trying to map where on NCAM polysialic acid is expressed. And it turns out there are 4 potential N-linked acceptors, but polysialic acid is really only found on these 2, which are adjacent to a fibronectin domain. So there's some recognition here by the polysialylotransferases to put on these long polymers. So if we look back at the differentiation, if we start with the human embryonic stem cell, there are 2 enzymes in humans that make polysialic acid, and very interestingly, If you go down the mesendoderm definitive endoderm pathway that I mentioned before, it is the enzyme PST, or also the ST8-4 enzyme. But if you differentiate down the ectoderm lineage, the other polycyclic transferase is responsible for synthesizing polycyclic acid. And if you knock out either of these and differentiate the cells, they will not differentiate. So polycyclic acid is absolutely required for differentiation from embryonic stem cells. And transcriptionally, if you look at PST in the definitive endoderm pathway that I just mentioned, it comes up after goosecoid comes up, and so there's evidence that that goosecoid can induce polycyclic acid through inducing PST, along with other genes that are responsible for early differentiation, and NANOG is basically turned off. So these enzymes are developmentally regulated very tightly. And just to show that this happens not just in embryonic stem cells, but also in developing organisms, this is a section from a chick embryo stained with E-cadherin, which stains this ectodermal layer, and then as differentiation occurs and mesoderm differentiates, the expression of polycyclic acid on NCAM shown here in green. So as these cells migrate down through the primitive streak and out again, you can see they turn on polycyclic acid on NCAM, and they lose some of the red stain, which is the ectodermal stain as well, the E-cadherin. So this regulation occurs in vivo as well. Now, if we want to understand the polysialome, initially NCAM was the only protein that was known that expressed polycyclic acid, and that seems to be true in the embryonic stem cell case. But now you can see now a list of other proteins where polycyclic acid is found, and these proteins were identified using that antibody that I mentioned before to target polycyclic acid and to pull out glycoproteins so that they could be identified by proteomics, and then the structures on them could be determined. So this is first example of using a reagent, in this case an antibody against polycyclic acid, to get at the proteins that express it. And this is one of those things, the harder we start looking, the more proteins that we find that actually express polycyclic acid. So it's an interesting collection of proteins. My graduate student, DJ Bernsteel, as part of his thesis work, mined the TGX expression library for the two sialyl transferases. You can't really see this too well, but there are a lot of other tissues that light up, but they're much more faint than these large ones. And then here are 4 different acceptors: NCAM, the neuropilins, and then also the chemokine receptor CCR7. You can't really see this well, but there's expression of these in many, many other tissues and cell types than we would normally think about. So, in conclusion, looking at polycyclic acid by pulling out specific glycoproteins using that antibody is giving us a way to focus in on the proteins that are expressing the polycyclic acid and what their structures are. So the polysialome, if you will. So here are the 3 linkages of sialic acid: alpha-2,8, 2,3, and 2,6. And this is the polysialic acid, for example. So what about these other sialic acid linkages? Could we target those to be able to pull those down? And there are lectins that have some specificity for alpha-2,3 and 2,6, but I will mention I'll mention a new reagent that's been developed. And so basically, if we focused on sialylated glycoproteins, really these are cell surface and secreted glycoproteins mainly that have sialic acid on them. So this is going to allow us to simplify the glycome and approach the sialome. We know specific functional molecules in the cell surface that express sialic acid, and we know that changes in expression can regulate function. And so would it be possible to determine the sialome if we had the right targeting reagent? And this shows a collection of N-glycans from mouse embryonic stem cells, and these differentiated cell lineages. This was done by Mike and Lance's laboratory. And so you can see all this collection of N-linked structures as well as O-linked. Almost all of these have sialic acid on them. So it would allow us to really ignore, if you will, the high mannose expression, the biosynthetic products inside the cell, and focus on the cell surface, which has sialic acid on it. And again, we know all these sidyltransferases, and we know that they change, for example, during differentiation. So that's an interesting approach. And so the reagent that I'm talking about is made by a small biotech company founded by Rob Woods at the CCRC called Lectins Bio. And the idea is to take enzymes that recognize specific glycans and modify by mutation those enzymes so that they bind and they don't cleave. So it's basically a lectin from an enzyme, because obviously the enzyme has to bind with high affinity before it cleaves, but in this case it doesn't cleave. And so one of the first reagents they've developed, or actually a couple of reagents, recognize all sialic acid, and then there's one that recognizes specifically alpha-2,3 link sialic acid. So that's going to be the hook to be able to look at the sialome. So here is a crystal structure. This is a bacterial neuraminidase that's been modified by lectins, and I'll just say I am not affiliated with lectins. The company, I don't have any stock in the company. I'm not an advisor, but I think it's a really interesting approach that could be used for us to go after the sialome. But you can see here, this is erythropoietin with 3 N-linked structures and an O-linked structure being bound by the reagent here. Of course, this can be biotinylated and used to pull out glycoproteins, or this could be trypsinized, and we could use it to pull out glycopeptides to be able to do the structures. This is a protocol developed by Mike's lab that he'll, I'm sure, talk more about, but anyway, the idea is that you could use the hook at a lot of different levels, either in the glycoproteins or glycopeptides, or to enrich for the glycans themselves. And I just want to provide one example of using a targeted approach. This is actually a 2008 study we did on invasive ductal breast carcinoma, where we took actual tissue, extracted the proteins. In this case, we used a biotinylated lectin, L-phytohemagglutinin, as I mentioned earlier, to be able to pull out glycoproteins that have specific beta-1,6 branches on them. And so the control here is matched breast tissue from the same patient. And the question is, do we see an enrichment? And really the purpose of the study was to identify specific glycoproteins that express these, and the question was, could we exploit those as potential markers? And we were actually able, I say we, this was really done by Mike and Kazuhiro Aoki, to be able to do structures on these sialylated beta-1,6 branch glycans that we pulled out using that approach. I won't go into all the data here. This is actually done quite a long time ago, but it was really one of the first examples of taking cancer tissue and doing glycomics on the tissue again, using this targeted approach. And of course, we were able to identify glycoproteins that were pulled down, that were specific in the cancer tissue for different patients' cancers. And one of those was periostin. And then we can actually verify these changes, this expression, using an antibody to periostin that we obtained commercially to pull it down, and then look for binding to LPHA to confirm that our scheme that I just described was really accurate, and we really see large differences in periostin with the LPHA branch on it in the cancer. So using the targeted approach, we can characterize the structures, but then if we have antibodies to specific receptors or transporters or whatever, adhesion molecules, we can verify the sialome focusing on specific glycoproteins in this way. So this is conclusion. This is, I believe, a viable method of focusing on the sialome as part of the glycome. And again, most of this work was done by Lance and Mike. You've seen here many years ago. They look a lot older now, as you can tell from Dr. Thiemeyer. And Kelly Moorman, who developed the PCR array. And as I said again, now this is all, the glycotranscriptome is all done by deep sequencing, but we can verify it with PCR. So I'll stop there. Thank you.